Hello students, welcome to EPG Pathshala. I am Dr. Maimoona Akhtar from School of Pharmaceutical Education and Research, Jamia Hamdard. Here we are going to discuss about the module Sensing Applications of Quenching from paper Atomic Spectroscopy. In this module, you will learn about the sensing applications of quenching like chloride sensing, fluorophores, intracellular chloride imaging, chloride sensitive green fluorescent proteins, the knowledge of quenching mechanism that is valuable for rational design of fluorophores with the desired sensitivity. In this module, we will also learn about the sensing application of quenching to molecular biology. We already know about the fluorescence, phosphorescence and the basic concepts, the use of fluorescence measurement, the use of quenching measurements. So let's have a brief discussion first about fluorescence quenching. We all know fluorescence is a kind of luminescence where the emission of photon is brought about by absorption of light and emission stops as the light is cut. The luminescence can be most conveniently defined as the radiation emitted by a molecule or an atom after it has absorbed energy to go to an excited state. There are two main types of photoluminescent processes, one fluorescence and another is phosphorescence. Fluorescence and phosphorescence are alike in that excitation is brought about by absorption of photons. The two differ in the lifetime, that is, the lifetime of fluorescence is shorter than that of the phosphorescence. Fluorescence can occur in all kinds of molecules, in simple as well as in complex, or in gaseous state also, liquid or solid, whatever system may be, or in biological systems even. So simplest example is vapor of sodium atom, wherein 3s is excited to 3p by absorption of the wavelength 5896 angstroms and 5890 angstroms. And the simplest example of biological luminescence is jellyfish. The fluorescence and phosphorescence is the ratio of the number of molecules that luminescence to the total number of the excited molecules. For highly fluorescent molecules such as fluorescein, the ratio approaches unity. The fluorescence has been widely used for study of chemical and biological reactions, to understand the dynamics of the biological system, and similarly, quenching of the fluorescence, which means inhibition or reducing the phenomena of fluorescence, has been widely used to study chemical and biological phenomena in research. And any process that can lead to reduction in fluorescence intensity is known as fluorescence quenching. This fluorescence quenching may be due to number of reasons, which we already know and have discussed in previous modules. For refreshing, the fluorescence quenching may be due to molecular rearrangement, the reaction of the excited state of the molecule. It can be due to collision of the quenching species with the quenching species, or it is loss of fluorescence by colliding with other ions or atoms. The energy transfer, energy charge reactions, or photochemistry may also be regarded as responsible for quenching of fluorescence. Fluorescence quenching has been widely studied both as a fundamental phenomena and as a source of information about various systems, including biological systems. The biochemical application of quenching are due to the molecular interaction that results in quenching, the molecular interaction of a fluorophore with the macromolecule. A wide variety of substances have been developed as fluorophores and as quenchers of the fluorescence, and we all know the best known collision quencher is molecular oxygen, which quenches almost all known fluorophores. Mainly two types of quenching processes are encountered, which are namely collision, which is a dynamic method, and static, which is a complex formation method. The collision quenching and static quenching. Application of quenching of fluorescence is in study of different biological systems and their function. For example, to study the diffusion coefficient of oxygen in membranes, or we can study the position of quencher in the membrane. Coming to applications of quenching of fluorescence, now I'll be discussing in detail the applications of quenching of fluorescence. We have already discussed the application which include oxygen diffusion in membranes, localization of membrane-bound tryptophan residue by quenching, quenching of membrane probes using localized quenchers, parallax and depth-dependent quenching in membranes, boundary lipid quenching, effect of lipid-water partitioning on quenching, quenching in micelles. The one which we'll be discussing in this module is sensing application of the quenching of fluorescence. The sensing application of fluorescence quenching has been used for a variety of analytes, including nitrous oxide, oxygen, and heavy metals. The use of quenching for sensing is exemplified by chloride sensitive fluorophores. There are a large number of fluorophores which are available, which have been used for their sensing application. Here we'll be exemplifying by chloride sensitive fluorophores. The chloride sensitive fluorophores, we all know that chloride is an important biological anion. It plays a role in neuronal processes, nerve conduction, fluid absorption, and maintaining the cellular pH. These chloride sensitive fluorophores have been extensively developed for their analytical and biochemical application. These fluorophores are based on acridinium or quinolinium structure with additional groups to modify their properties like solubility or sensitivity of fluorophore to quenching. Usually chloride quenching is collisional type of quenching with some exceptions, as can be seen in figure on next slide. The figure shows Stem-Vollmer plot for quenching of N-methyl quinolinium iodide by chloride or iodide. We can see in graph the quenching of fluorescence by chloride is more efficient than that of iodide. The equivalent decrease in intensity and lifetime shows that quenching is collisional and complex formation does not occur. We can see the amount of quenching by using iodide is not very significant compared to the chloride. Electron transfer from the anion to the fluorophore seems to be the most probable mechanism of quenching, and it suggests that the quenching efficiency should depend on the oxidation potential of the quencher. Accordingly, the iodide has the highest quenching efficiency as it gets easily oxidized compared to the other halogens. When compared to the stability, the fluorides are more stable than iodides because of their high electronegativity. It is difficult to get them oxidized, hence there is much less efficiency in quenching. Also, even though the diffusion coefficient of chloride and iodide are similar, experimentally has been observed that iodide are more efficient in quenching than chloride and fluoride. It is believed that iodide is more strong quencher or strongly quenches than the chloride. The other figure shows biomolecular quenching constant for N-methyl quinolinium iodide and several quenchers. The quenching efficiency is highest for iodide because of its easy oxidizing nature. The quenching of N-methyl quinolinium iodide by using various quenchers, bromine, chloride or acetate, we can see due to the fluorine, the quenching is least, followed by acetate ion, then chloride ion and bromide ion, and the highest is seen by the iodide ion. The quenching efficiency for a charge transfer mechanism is expected to depend on the energy charged for the reaction. For a charge transfer process, the free energy change is given by delta G in electron volts, which is equal to the energy of the oxidizing state less energy of the singlet energy of the fluorophore, where you can see E ox is the oxidation potential of the electron donor, that is the quencher. E reduction is the reduction potential of the electron acceptor, the fluorophore. E00 is the singlet energy of the fluorophore. C is constant, that accounts for the energy release due to the charge separation interaction of the charge with the solvent. With increase in oxidation potential, the value of the free energy delta G also increases. That it becomes less negative and as a result less efficient quenching. The above equation does not correlate with the quenching efficiencies absolutely because other effects like electron exchange and heavy atom effects also have their effect on quenching. Although for this class of probes, charge transfer is the dominant mechanism for quenching by halides, other effects also get involved. Knowledge of quenching mechanism is valuable because rational drug design of fluorophores with the desired sensitivity to halide depend on it. That means if you want to design a fluorophore for specific use, you have to have a knowledge before the quenching mechanism. For example, substitutions on the quinolinium ring that decrease the affinity for electrons are likely to decrease the quenching efficiency. So therefore, you need to keep in mind the type of substitutions you need to add on any system, whether they increase the quenching efficiency or may decrease the quenching efficiency. Intracellular chloride imaging. The chloride-sensitive fluorophores have also been widely used for imaging the local chloride concentration and measurement of the intracellular chloride concentration. Because chloride concentration is very important in functioning of the cells, especially conduction of the signals. Due to photobleaching and the unknown concentrations of probe at each position in the cell, the use of fluorescence microscopy for imaging is difficult. In contrast, calcium and other cations, collisionally quenched probes, do not show spectral shifts. In earlier days, direct wavelength ratiometric probes for chloride were not available, but the knowledge of the mechanism of chloride quenching has helped in the design of these probes. This consists of linking two fluorophores, one which is quenched by chloride and one which is not sensitive to chloride. Here is an example of direct wavelength ratiometric probe for chloride. This probe contains two quinoline rings. The methoxy substituted ring on the left readily accepts electron and is quenched by chloride, whereas the group on the right side of the quinolinium, which is an amino group, does not accept electrons from chloride and therefore is not affected by it and hence no quenching. Here we can see two figures. One is emission spectra of MQXY DMAQ and effect of increasing concentration of chloride on its spectra. The graph shows emission spectra in the presence of increasing chloride concentration. The shorter wavelength emission is quenched. You can see there is decrease in the intensity when increase in the chloride concentration, but the longer wavelength, that is wavelength at roughly 550 nanometers, is not sensitive to chloride, therefore no effect on its intensity could be observed. Examination of the spectra in figure 4 suggests that there will be red from the MQ to the DMAQ ring. In this probe, some energy transfer occurs which reduces the lifetime of the MQ ring, methylquinolinium ring, and makes it less sensitive to quenching by chloride. However, the red efficiency is modest so that the MQ ring remains sensitive to chloride. The sensitivity of a probe differs in a pure solution than its sensitivity when used in intracellular environment. Therefore, the chloride-sensitive probes were calibrated in cells by changing the intracellular chloride concentration. This is done by exposing the membrane permeability of the 3T3 fibroblast cell using valinomycin and other similar compounds, followed by exposing the cell to different chloride concentrations. This is reflected by decrease in intensity of the short wavelength MQ emission at 450 with the increase in chloride concentration, as we saw in figure 4, and the intensity of the longer wavelength emission at 565 nanometers remains insensitive or unchanged. The wavelength ratiometric chloride probe have been used to measure local chloride concentration in CHO cells. by collecting the intensity image at 450 and 565 nanometers. The intensity at 450 nanometer is sensitive to the local chloride concentration, whereas the image at 450 nanometer is not sensitive to the chloride concentration. Therefore, the ratio of the emission intensities of the two can be used to determine the concentration of chloride in the cell. These images revealed uniform concentration of chloride in these cells with a somewhat lower chloride concentration in the nucleus. Here we can see the fluorescence microscopy imaging of the CHO cells with chlorine sensitive probes or chloride probes. The fluorescence microscopy image of CHO cells labeled with the chloride probes and intensities at 450 and 465 nanometers are color coded for clarity. The image on the right are the chloride concentration calculated from the intensity. By seeing the difference between the two intensities, the ratio will give you the exact concentration of the chloride in the system. We have already discussed about green fluorescent proteins in previous modules, how they have been used and different types of fluorescent proteins developed. Here also green fluorescent proteins and its mutants are widely used to study gene expression and as sensors. A yellow variant of green fluorescent protein, which is also known as YFP, is known to be sensitive to chloride. That means it is very sensitive to change in concentration of the chloride. The intensity of the YFP H148Q decreases progressively in response to the added chloride in a manner and concentration range that suggests collisional quenching. The right hand side you can see the graph which results or which shows with effect of pH on the intensity of the fluorescence of YFP H148Q in presence of sodium chloride. With increase in pH, the change in the fluorescence is visible. The extent of quenching depends on pH, which is not expected for collision quenching. The reason for this is that the chromophore in GFP is buried in beta barrel structure of the protein, which is shown on the right hand side. You will see two chromophores, one in the inside, within the barrel of the protein and another at the top. And you can also see the green structure, the native chromophore, which is near to the blue surface, which is an iodide ion. These observations suggest that the chloride sensitivity of the YFP is not due to the collisional quenching. It is rather the inaccessibility of ions to the chromophore, because this does not allow free access to the diffusing chloride or iodide. This figure shows the absorption spectra of YFP H148 in presence of sodium chloride at pH 6. Binding of chloride is thought to stabilize the right side structure in the upper panel. An iodide or chloride next to the chromophore might result in static quenching. However, static quenching by chloride is not the reason for the chloride sensitivity of yellow fluorescent proteins. It appears that the quenching of fluorescence of YFP is due to an induction of change in the structure of the chromophore by chloride. The chloride is believed to bind near the chromophore in contact with the imidazoline oxygen atom, which forces the shifting of the equilibrium to the left. The large amount of chloride quenching at lower pH is consistent with protonation of the phenolic oxygen upon binding of chloride. Hence, the apparent quenching of YFP by chloride is not due to collisional or static quenching, but instead due to a specific binding interaction which changes the structure of the chromophore, therefore rendering it non-fluorescent. A wide variety of fluorophores are available that can be used for intracellular sensing. It is difficult to use these probes in an intracellular environment because they are likely to bind macromolecules that can shift the analyte calibration curve. GFPs and its variants are promising intracellular sensors because the chromophore is buried in the J3 barrel and unable to interact directly with the biomolecules. Due to this reason, YFP H148Q has been used to measure the intracellular pH of Swiss 3T3 fibroblasts. An advantage of GFP is that the probe is synthesized by the cell itself and not added to the cell. The fibroblasts are transfected with the gene for YFP H148Q, which are a bright green fluorescence. The response of the protein to pH and chloride has been tested using ionophores to modify the internal ion concentration. When excited at 480 nanometers, the emission intensity decreases with increasing pH and decreases with increasing chloride concentration. These remarkable results show that it is now possible to insert genes that cause the cell to synthesize their own fluorophores, and the genetically engineered protein fluorophores can be designed to have sensitivity to a desired analyte. Sensing application to molecular biology. Many advances in the development and use of sensing for DNA analysis have been during the past decade. A number of techniques make use of quenching. A typical situation is the need to detect a target oligomer of known sequence in a sample containing many sequences. The detection of target oligomer is usually accomplished by synthesis of a probe oligomer with the complementary sequence. The probe oligomer also contains bound fluorophore that displays spectral changes upon hybridization with the target oligomer. The spectral changes can be due to red, intercalation, or quenching. Release of quenching upon hybridization. This figure shows the release of the quenching or when there is hybridization of the molecule. One approach to target detection is to change the extent of quenching upon hybridization. In this case, the fluorescein label is quenched by a nearby pyrene residue, and in the absence of target DNA, the fluorescence is quenched by the pyrene. When the same is bound to the target sequence, the pyrene interacts or intercalates into the double helical DNA, resulting in several fold increase in the fluorescence emission. This approach is general and could be used with a variety of fluorophore quencher pairs. Molecular beacons in quenching by guanine. Many fluorophores are efficiently quenched by the nucleic acid base guanine, probably due to photo-induced electron transfer. The other bases are much less efficient as quenchers. In next slide, we can see a Stern-Walmer plot for DGMP as a quencher with 4 fluorophores. The upper left panel shows you the structure of 4 fluorophores, that is JA242, MR121, R6G, and TMR. And right side panel shows you the quenching of all the fluorophores with guanine monophosphate. The lower panel shows the quenching of fluorophore MR121 by all the nucleotide phosphates, that is guanine, adenine, cysteine, and thymine. From these results, it can be seen that quenching by guanine monophosphate is not limited to single fluorophore, but multiple fluorophores can be quenched efficiently with it. Among the 4 probes studied, MR121 has been reported to be most sensitive to quenching by guanine monophosphate. In the efficient testing of all the nucleotide monophosphates, guanine monophosphate against MR121 has been found to be the most efficient one. This quenching property of guanine was used to design a molecular beacon. The fluorophore JA242 was positioned at one end of the beacon, and the other end was terminated with the several guanine residues. The residue quenched the fluorophore when in the folded state. The emission intensity increases about 5 folds when the beacon is unfolded and hybridization with the target sequence takes place. The right side panel, you will see the absorption and emission spectra of a molecular beacon based on guanine quenching of the JA242. Binding of substrate to ribosomes. We already know guanine has been a very good quencher of various fluorophores. So quenching by guanine has also been applied to structural studies of RNA. Compared to DNA, RNA is flexible because it's single-stranded and frequently forms base paired region within a single strand of RNA. The single strand of RNA, like peptide chain, folds and can adopt well-defined three-dimensional structure in solution by folding. Ribosome is one part of the structural DNAs that displays catalytic enzyme-like behavior. That means they can catalyze reactions. The catalytic properties of these highly structured RNAs was first reported in 1982, showing that certain RNA display autocatalytic activity. Numerous examples of studies related to ribosomes and their catalytic behavior are reported in literature. One example is a hairpin ribosome that cleaves single-stranded RNA. The observation of quenching by guanosine nucleotide suggested the use of guanine quenching to study substrate binding to ribosome. The substrate contained a fluorescein residue covalently linked to 3' end. Herein you can see the structure of hairpin ribosome, which has folded and form a double strand by base pairing, and we can also see the fluorescein attached to 3' side of the ribosome and the guanosine attached to the 5' of the chain. The substrate binding region is the region adjacent to the substrate. The hairpin ribosome contains a guanosine residue at the 5N end. The fluorescein emission is quenched when the substrate nucleotide binds to the ribosome. The quenching also occurs when the fluorescein labeled substrate binds to the substrate binding strand that contains 5N guanosine residue. The guanosine residue is required for quenching, and the emission of fluorescence is unchanged in the presence of the substrate binding strand without a 5' terminal guanosine residue. The example shows how fundamental studies of nucleotide quenching have found useful application in modern biochemistry. Herein you can see the emission spectra of the fluorescein labeled substrate analog in solution and when bound to hairpin ribosome or the guanine containing substrate binding strand. You can see the difference in the intensity of the fluorescence of the two. In this module, we have learned about the sensing application of quenching, because we already know about quenching, application of fluorescence, application of quenching. We have in this module specifically stressed upon the sensing application of the quenching by exemplifying using chloride sensitive fluorophores. We also discussed mechanism of rational design of fluorophores with desired sensitivity, how the position of chromophore affects the mechanism and sensing ability of the ion quencher. In this module, we also learned about how hybridization leads to release of quenching. That means if you have a quencher and a fluorophore on the same strand, by hybridization, the intercalation of the quencher takes place and the quenching is released. And what are the other molecular beacons? We also discussed that they are making and working, and the use of ribosomal RNA in sensing application. Thank you.